So to identify bacteria in the lab, we have to, we can use differential medias that contain different substrates, different enzymes, to determine how the bacteria react in that particular environment. And how they react will give us a color change, and then from that color change we're able to interpret different biochemical and metabolic activities that take place within the bacteria. The next attribute that helps us to characterize bacteria is that of their pathogenic nature, which is also known as pathogenicity. And many pathogens are able to elicit disease in a host because they possess specialized structures. And those structures are things like a capsule, pili or endotoxins, or exotoxins and exoenzymes. Now, capsules enable bacteria to evade mechanisms of the immune system that can be used to get rid of bacteria from the host body. But the capsules can't be digested by immune cells, and so this provides extra protection for the bacteria, making it more pathogenic. Pili help to allow bacteria to adhere to tissue, so this also enhances pathogenicity of the bacteria. And then endotoxins are toxins that are released whenever a bacterial cell is actually degraded or dies, kind of like that lipid A we were talking about when we talked about the lipid polysaccharide. Or you can have bacteria that secrete what's called an exotoxin, and an exotoxin, an example of that would be something like botulinum toxin or Shigella toxin, or well, those are kind of the key ones. But those exotoxins are released and they make the host tissue very, very weak and start to break it down. And then there's also exoenzymes, which are kind of the same as exotoxins, except for exoenzymes are released and they start to break down different substrates. So like the bacteria in your mouth exhibit an exoenzyme called amylase, and that helps us to break down starch molecules. So these exoenzymes and exotoxins can also be used to help bacteria become very invasive and start to destroy host tissue. Pathogenicity is generally tested by taking a bacterial organism and injecting it into a mouse or a cell culture, and then seeing how that bacteria operates or impacts those particular culture models. And some specific types of pathogenic bacteria are Neisseria meningitis, Salmonella typhi, Shigella subspecies, Vibrio cholerae, Yersinia pestis, Treponema pallidum. Neisseria meningitis causes, like the name implies, meningitis. Salmonella typhi is basically salmonella infection with a lot of gastrointestinal complications. Shigella subspecies is basically gastrointestinal complications, very similar to salmonella. Vibrio cholerae, again, gastrointestinal complications. You're going to get, when I say gastrointestinal complications, you can kind of correlate that with having diarrhea. You can also have a lot of vomit and a lot of projectile fluid that comes out of a patient suffering from these particular organisms. With Yersinia pestis, this is an organism that causes the bubonic plague, so you can have giant boo-boos start forming all over your body, which leads to necrotic tissue. And then you also have another bacteria called Treponema pallidum, which I've talked about before. Remember, this is the organism that causes syphilis due to twisting motility of the organism. So the key thing about this is that for us to characterize certain bacteria, we base it off their pathogenicity. The next attribute, which is our kind of last attribute, is going to be that of genetic composition. And genetic composition is where we use simply molecular techniques to understand an organism's RNA or DNA. We look at simply comparisons between different species to figure out if the DNA or the RNA has been conserved in some areas, and this will allow us to understand kind of the evolving nature of different bacteria. And what's great about bacteria is they replicate so fast that you could essentially have evolution within the span of a week. That's actually observable inside the laboratory setting. So it's very neat to see when you compare the genetic materials of different organisms to each other. But you can also look at the ribosomal RNA. Remember we talked about the ribosomal subunit being the 16S. If you take that RNA and you sequence it and you compare those sequences among different species of bacteria, you might see a little bit of evolution and be able to understand the degree of relatedness between the organisms, because the 16S ribosomal RNA is very much conserved. So anytime there is evolution at some point in that ribosomal RNA, it's very interesting because you can pinpoint where the mutations are. So that's going to lead us to kind of looking at some unique bacteria that are out there. There's some groups known as rickettsias, chlamydias, and then mycoplasms, which we've talked about. These guys are not typical bacterial cells, so we're not going to see typical coccus or bacillus-shaped bacteria here. The rickettsias and the chlamydias contain, are basically gram-negative. Regarding their cell wall structure, so that means they have a thin layer of peptidoglycan, but they have to live inside of another host. Okay, so they're considered obligate intracellular pathogens, and they can't be produced in vitro in artificial culture media. So we actually have to have host tissue if we were to culture rickettsias and chlamydias cells inside of a laboratory. And rickettsias have what we refer to as leaky membranes, which means that there's not a lot of structure to them, which is probably why they're obligate intracellular pathogens. And chlamydias are known as energy parasites, which essentially means that they like to use ATP molecules that aren't going to be produced by their cells. They're actually going to be generated by the host cell that they're feeding off of. And here's some images of Rickettsia prowazekii, which is an organism that led to louse-borne typhus. And you can see the organism situated right here. Okay. The mycoplasms, which we talked about, remember these are the organisms that have to be stained special using the acid-fast stain. They don't have the cell wall. So we refer to these bacteria as being pleomorphic because the absence of the cell wall causes the shape of the cell not to be very uniform. And so you actually see different cell shapes underneath the microscopes if you're looking at a mycoplasm under the microscope. And this is also one of the smallest of all the cellular microbes that are out there. So pathogenic mycoplasms typically can cause pneumonia as well as genitourinary infections. So these can have some very implicit pathogenic properties to them and induce disease. They, again, lack a cell wall and can be very resistant to drugs such as penicillin. So mycoplasms aren't anything that's fun to work with. And here's Mycoplasma pneumoniae, which you can see here on this slide. Here's the pleomorphic nature, where you have these round shapes and then these long broad shapes on the same slide. In most bacterial specimens, you wouldn't see that. Photosynthetic bacteria are also another kind of unique bacteria because they contain chlorophylls and they undergo the process of photosynthesis to help generate sugars for the cells. So bacteria that fall in this category are purple bacteria, green bacteria, and cyanobacteria. They all will rely on light as an energy source, but they use the source differently. The purple and green bacteria, they don't produce any oxygen. The cyanobacteria are the only bacteria that are going to have a waste product of oxygen, which is pretty important because if you think about evolution, a lot of studies have indicated that cyanobacteria were precursory or forerunners of the oxygen movement, which led to the rise of plants in our evolutionary tree. So very important role. Two things to note about this is that there are oxygenic photosynthesis process and anoxygenic photosynthesis process. So when oxygen is being produced, we call it oxygenic photosynthesis. So that's what the cyanobacteria will be doing. And then when oxygen is not produced, we call it anoxygenic photosynthesis, which is what the purple and green bacteria undertake. And then lastly, we have an archaea group, which archaea means ancient. So these are among the older lineage of our prokaryotic organisms. And they were discovered in 1977. As I said many times, these organisms are still prokaryotic, which is why we're discussing them. But they need to live in very extreme environments in order to survive. So environments where there is very low acidity, or a very high alkaline condition, or where it's extremely cold, or where it's extremely hot, or where it's extremely salty. But these are the extreme guys. So they're also known as extremophiles because they live in extreme environments. So many members of the archaea group can withstand a lot of conditions that aren't very conventional for most organisms. Archaea bacteria have cell walls, but their cell walls lack peptidoglycan, which makes them different from bacterial cells. Because remember, bacterial cells, their key feature is that they have peptidoglycan, either a thick layer or a thin layer. But for the most part, archaea bacteria are going to be absent of peptidoglycan, which is a definitive characteristics of their structure. And here's an example in Maritimus, which is a archaea group, archaea organism that resides inside the oceans, and it uses carbon dioxide as well as ammonia to generate nitrite, as well as organic carbon to be released, and fits in with the global nitrogen and global carbon cycle. Okay, so this is going to conclude this unit on today's lecture. So I hope you learned a lot about this, and if you, as always, if you have any questions, please don't hesitate to contact me. All right, you guys have a great day.